talking about Tay-Sachs disease, the cause, the genetics, and the impact on the Jewish community. The primary sources that I've cited are the mystery of Tay-Sachs as a Jewish disease, Tay-Sachs screening motives for participating, and knowledge of genetics and probability, and efficient and precise editing of Tay-Sachs disease model in a rabbit by prime edits. Tay-Sachs disease is a disease that is very detrimental and lethal, specifically for the Ashkenazi Jewish community. The disease is passed on when two parents are carriers, and the child inevitably dies between ages 2 and 5. The disease causes a buildup of GM2 ganglioside, which is usually removed from the neurons by an enzyme called hexosaminidase A, or hexa, and the mutation occurs on the 15th chromosome. Over here, you can see other very serious diseases that occur on the 15th chromosome. In this visual, you can see the difference between normal neurons and Tay-Sachs neurons. You can see how enlarged the Tay-Sachs neurons are, and that's due to the buildup of the GM2 ganglioside in the neurons. The Tay-Sachs neurons are essential for, the Tay-Sachs neurons that are affected are essential for human survival, and it is truly one of the worst diseases that affects the Jewish community. But among the other ones, Marfan's is also very significant. There are many theories about how the Ashkenazi community kind of got stuck with Tay-Sachs disease, but the most validated one is genetic drift, which is when a community gets significantly reduced in their population. Now, for anyone who needs background, Ashkenazi Jews are ones specific to the regions of Russia and Western Europe. Now, after the diaspora from Israel, they fled there and usually congregated into very small communities. Multiple tragedies have occurred, such as the Bar Kokhba Revolution. Oh. Huh? I think yeah. The Chmielnitsky massacres and the Holocaust that have significantly reduced the population of the Ashkenazi Jewish community. The first experiment that I'm going to be looking at is the motives for participating in knowledge of genetics and probability. This experiment was carried out by Dr. Michael Kavak in the mid-80s. Dr. Michael Kavak has almost eliminated Tay-Sachs from the Jewish community through screening. Whenever a Jewish family wants to have a child, it is generally the rule that they will get screened to see if they are a carrier for Tay-Sachs. When he started his screening and testing, he also performed a social experiment on participants, seeing how they felt in general. Now, in the 1980s, biology had only had genetics required in the course for about 20 years. So most adults that were dealing with this problem didn't know about genetics and didn't know why their kids could be carriers for Tay-Sachs. As you can see, he asked multiple questions, such as what is the frequency of Tay-Sachs in Jews? What is the carrier frequency? What's the chance that your child may marry a carrier? What is the chance of your sibling being a carrier? Can your child be a carrier? What is the chance that your child will be a carrier? And is there any chance that your child may have Tay-Sachs disease? The goal of his work was to make it so that screening for Tay-Sachs would become the rule rather than the exception. And his testing revealed the need for more knowledge of genetics, not just among society, but among physicians who didn't know much about the disease. Getting into the rabbit experiment. As I've stated, Tay-Sachs is a progressive neurodegenerative disorder due to the deficiency of hexosaminidase A, or hexa. This SNP that it lies on is RS387906309, which is the one most directly correlated with Tay-Sachs disease in Ashkenazi Jews. 80% of Ashkenazi Jews who have Tay-Sachs have it because of this SNP. The genotypes are negative TATC, and TATC. This is the one that is Tay-Sachs disease, and this is carriers. The insertion of TATC in exon 11 is the cause of Tay-Sachs. There are 5 different variants. The first is classical Tay-Sachs disease, which causes a death between 2 and 5, juvenile onset, which is fatal by 10, chronic GM2 ganglioside, which continues into adulthood, adult onsets, which starts in one's 20s and 40s and is fatal, and hexa-negative allele. And hexa int TATC introduces premature termination codons in exon 11, which led to the deficient activity in the rabbits. The phenotypes produced in the rabbits were increasingly frequent head raising, convulsions, abnormal gait with decreased sway length, clasping of the limbs, increased cervical lordosis, muscular fibrosis, and enlargement of preneural space. All of these, if you think about it, the defects of the nervous system that Tay-Sachs cause would be the cause of all of the deficiencies in the muscles. So just to explain this figure here. Figures A, B, C, D, E, and F, and G all pretty much show the effects of the CRISPR technology insertion into the rabbits. So figure A shows the base insertion efficiency. This is the efficiency in embryos. This is the PE activity in grown rabbits and embryos. And then these figures show the base editing and also other frequencies. This shows the protein insertion, but the main figures I'm going to focus on are J, K, and L. J shows the cervical lordosis in the rabbits and the clasping of the limbs and the legs. K shows the myopathy with fibrosis from the inflammation, and I shows samples from the hippocampus. If you look at this right here and compare it up here, they're very similar. The rabbits' neurons can be quickly compared with the human neurons, and the reason rabbits were chosen for this experiment is because mice weren't affected the same by the disease, which creates a phenomenon that's revolutionary, which is the idea that rabbits could be used to develop a drug to combat Tay-Sachs disease in the future, which would take even more weight off the Ashkenazi Jewish community, similar to Dr. Kavak's experimentation. So if you look here, the phenotypes can also mirror people with chronic adult gangliosidosis with the limb girdle weakness, the ataxia, and the progressive neuromuscular weakness. And this study is so amazing because it verifies the feasibility of base conversions in rabbits, which means that this could generate treatments in the future. The inflammation with the fibrosis through staining is really what solidified me to choose this experiment, just because of how revolutionarily similar it is to the humans. And hopefully this study could be used to maybe even wipe out the disease from Ashkenazi community in the future. And even from Dr. Kavak's, it's said now that because of his experiment, there's more Tay-Sachs outside of the Jewish community than inside of it, which is already amazing. But if we could use these experiments here to wipe it off of the planet and develop a drug, it would be revolutionary, and that's why I chose these experiments.